Thank you very much for this really nice introduction. Thank you very much for the invitation to come to participate at this conference. It's a great honor, to be here at this, inaugural meeting. And I was already very impressed by the first speaker, so I will do my best to follow up. So I am a little bit different breed. I'm a basic scientist. I'm a Ph.D. scientist. So we're really trying to understand on a molecular level, how does exercise affect the brain? And I will also more focus on cognitive function. So how to preserve cognitive function in the context of aging and in the context of neurodegeneration. And I will bridge the human data that we know with some of the really mechanistic study that we're doing in the lab. So. All right, and again, I also have some disclosures. I hold patents, and I'm a co-founder of a company that is making use of those patents to develop novel therapeutics. All right, so coming from the almost a historic perspective now, if you think about exercise, it's something that affects the whole body, right? When you exercise, you improve your muscle mass, your muscle strength. You're doing something good for your vascular system, like your blood vessels. You're doing good things for your metabolism if you think about type 2 diabetes, if you think about weight control. It has direct effects on your heart. You can actually grow your heart. You can improve the cardiac capacity. This can give you, a protection against stroke and other vascular benefits. And all of this plays a role of how exercise is good for your brain. So there are these two mechanisms. The one is what we say you're reducing these modifiable risk factors such as body weight, type 2 diabetes, hypertension, and then there are these intrinsic factors in the brain where the exercise is rewiring the brain directly, which is another part I'm going to talk about. So just a very brief refresher, when we talk about exercise, we actually distinguish exercise and physical activity. Physical activity is everything that you do when you move around in daily life, going up the stairs, taking care of your garden, doing housework, and even that counts. So even that counts towards some fitness goals or daily physical activity goals. Exercise is like structured, planned type of physical activity to maintain or improve physical fitness. So that's kind of a next level. But as I said, my take home is both of them count. And then in exercise, we distinguish between two kinds. The one is the endurance or aerobic exercise. The classic one is running, but you could also do hiking, what's one of my favorites, biking or swimming. And then we have the resistance exercise. This is mainly to build muscle strength. The classic one is weightlifting, but there also there's like a lot of kinesthetic work now, or people doing yoga where you use your body weight to strengthen your muscle. And also in between that, another important goal is increasing flexibility and coordination, because again, the prevention of fall and aging is one of the main goals. And what is important for this talk is the evidence that we have for improving or keeping cognitive function. The strongest is we have on endurance exercise, on aerobic exercise. Again, this is not to make the point that devo exercise is not important. You have to keep up your muscle strength, or your patients have to keep up their muscle strength. They have to keep up their coordination and flexibility to be functional in daily life and to prevent falls. But if you think more about the thinking part, this is really where the aerobic exercise comes in. All right, so this is recent data from colleagues in mine at MGH. What we did is, this was an intervention study. So they took middle-aged people that had a sedentary lifestyle. And then they said, okay, we don't really care what kind of exercise you do, but you have to get at 50 to 75% of your maximum heart rate. They used like a Fitbit type device to measure that. And this is for 30 minutes every day. And then they did this for 12 weeks. And then what we can see is the training effect that was there was an improvement in VO2 max, which is just a fancy way to say how is your cardiac output and cardiorespiratory output improved. So yes, there was a training effect. But the interesting part is if you look at cognitive function, this was done by colleagues of mine. They used a whole battery of tests to look at different domains of cognitive function. There was also an improvement there, just by this 12-week intervention, by people choosing their own modality of exercise. And another important part is this improvement of VO2 max correlated to the improvement in cognitive function, which means the more you were able to push yourself, the more you improved your training effect, the more impact that had also on your brain function. And this is an observation we see in almost all the parameters we can measure in science. So now thinking about Alzheimer's disease, there is really good data out there that having an active lifestyle prevents the onset of dementia. This is data from the Rush Memory and Aging Project. This is just one study. And what they did is they observed people for 5 years. They asked them to record their daily activity, and then either you're in the top 10 or the bottom 10 percentile of daily physical activity. And if you're in the top 10%, you reduce your dementia risk by 50%. That's a pretty big reduction. And then looking at other studies, this is down there from an overview, they found that the maximum benefit was almost like if you do 10,000 steps a day, or roughly 9,100. But even if you get yourself to get to 3,800, close to 4,000 steps, you can reduce the risk of dementia. And if you think about a very sedentary patient population, 3,800, 4,000 steps, for some of them, that's already a lot. And I think giving them this reachable goal is a great motivator versus telling them like, oh, yeah, you should just go run a mile a day, or you need to do your 10,000 steps, otherwise it doesn't matter. These are other studies, just to make the point, a lot of different studies have looked at this effect. And the studies where they didn't see a reduction in the onset of dementia or slowing down of the cognitive decline that happens, most of these studies either didn't have an aerobic exercise component, or they did have it, but the participant actually didn't improve much there was not much of a training effect. So either that their training was too hard, they couldn't do it, or the training was not hard enough, and that's why there were not improvement. And CFR, that's cardiorespiratory fitness. All right, so coming back to more on a level, how can we monitor this in the brain? How do we know it does something to the brain? And this is one of the really nice first studies from Irisin et al., where they used MRI imaging to look at the size of the hippocampus. The hippocampus, as you probably know, is an important part for learning and memory in the brain, and it's also one of the areas that responds the strongest in exercise. So they had an aging population where they had an aerobic exercise intervention several times a week. They did MRI scans at the baseline, 6 months and 1 year, and they could see that the size of the hippocampus was increasing by 2% if you did the exercise, but you actually lost 2% of the hippocampal volume in the non-exercise group. This is not that all the neurons are just like, you're growing new neurons, but you are preserving synapses, you're preserving part of the neurons. So I think, again, that's a very strong effect. And another study from some colleagues, they actually looked at cerebral blood flow and angiogenesis in the brain. So again, you can do this with imaging-based technologies where you use a contrast dye and you can look at how much blood flow is coming in and out. And again, these correlation graphs show you the more you do fitness or the more you improve your cardiorespiratory fitness, the more blood flow you had in the brain and the more your executive performance also changed. And this little picture, this histology image here, this is from a study in, I think it was mice or rats. This is a part of the hippocampus right here. The red part is the blood vessels. We can stain these blood vessels. And you can see the mice that were running, they actually increased the perimeter of their blood vessels. So you're literally growing new blood vessels in the brain when you do exercise. Coming back more to the mice work, which we are doing, mice are actually a good mouse model to study exercise. They are responding to this very well, so it's actually a good model. This was one of these first studies where we looked, like my colleague Henriette van Praag did this like several years ago at Rusty Gage's lab, where she showed that even if you have young wild-type mice, if they do a training, they can improve their cognitive function. So what you do is, she added these running wheel cages to the mice. So we put them in the cage. The mice go on there. They love to run at night. They run several kilometers at night, 5 to 7 kilometers a night. And then you can see, you can test them after this running intervention in what we call the Morse water maze. So it's a water tank where there's a platform in there and the mice have to find it. Mice don't like to swim, they want to get out. And they have a training period, and the smarter the mice are, the quicker they learn this. And this is what you can see with these two graphs. The runner mice learn it much faster, and then later when you ask them, hey, do you remember where the platform was? Again, they pick out the right area of where the platform was, but not the sedentary mice. For aged mice that holds up, this is even like more striking, the contrast. This is when these are mice that hadn't had exercise a day in their life until they were very, very old. And then they were exercised, I think, for a month or two with these running wheels. And they did again this task. And you can see the old mice, they need a long time to remember how to get out of this pool. The ones that are running, they know exactly, this is why I got in, this is how I get out. So to the question, does it make sense, even if I haven't done much exercise in my life, does it make sense to start now? This data says very strongly, yes, it does. This is an imaging study that I did together with Dr. Christiane Wrann, who is at the Martino Center. So the idea was, how do we bridge, when we do our research, human and mouse data? In mouse, we have the advantage that if you want to collect any brain tissue at any time, we can do that. But for humans, obviously, to tell someone, can you do an exercise intervention, and then we're going to do a serial brain biopsy to look at your genes. I don't know if you get volunteers for that, but we wouldn't get it through the IRB approval process either. So we are mainly left to like using more non-invasive imaging technologies. But the good news is imaging technologies have improved so much that you can go down maybe not to the single cell level, but you can go down to the microstructural level. So my colleague, he is an expert in using the specific imaging technology, which is called DTI, or diffusion tension imaging with MRI, where you can look at, and I'm going to make it very simple because I'm not the physicist here. So basically, you can look at how the atoms are moving around, the water atoms. If you have an organized structure as in white matter, they don't have that much movement. In gray matter, they can go wherever they want. And that's basically what you can image. And here, this is a picture of a heart where you can remodel all the myofiber. All these are all the fibers in the heart, and you can really nicely see how you get these high-resolution images. So we did this in mice. We said, let's use the same technology we have in humans, and can we see the things in mice? And if that's true, then we can translate back and forth. Again, young wild-type mice, running exercise, looking after 1 month, looking after 2 months. And the first thing we found is we also saw this increase in hippocampal volume. This is this graph. So this is a mouse brain. Here, this little red insert, this is where the hippocampus is in the mouse. And if you measure the volume, it's going up. So again, one thing that does translate from mice to humans. And then we looked at, this is called the fractional anisotropy. That's the fancy word for the output, what the DTI imaging is doing. And what it does show is you get a more organized microstructure. So probably it's either more fibers, more synapses, or a reorganization. So is this a training effect we can monitor in mice, and we can translate that to humans. So this is a pretty important step in our work, because eventually whatever we find in the mice, we need to translate this to humans. We also looked in the hearts of these mice when they were doing the running exercise, and we also see a very nice remodeling of the cardiomyofibers with exercise, showing that there are, as we call it, like functional, histological, morphological correlates of the exercise. So we can actually measure in the tissue what exercise is doing. So coming back to some of the special mechanisms, how exercise makes your brain work better. And one of the most fascinating parts that I find is where it can actually make you grow new neurons in your brain. And this is really special because in the adult brain, there are only 2 areas where you can regrow new neurons. One is the neurons that help you with the olfactory function, with smelling. And the other one are actually in the hippocampus, where they help you with thinking, executive function. So this is images from mice. Again, this is the part of the hippocampus where these stem cells are. It's called the dentate gyrus. And what they did with these mice, they injected a substance called BRDU. It gets incorporated in cells that are dividing. So every time the stem cells are dividing, you get one more black dot. And you can see control mice, there are a couple of them running. You get an increase by 5 times, roughly, more neurons. And then you can do more fancy stainings to prove that these black dots are, in fact, actually neurons and not just some other cells that you might or might not care about. And here you can even trace these neurons with using a reporter virus. You can see these are growing up to become really beautiful neurons. They integrate in the circuits. They can talk to their neighbors. So, and there's many, many studies now that have shown the importance of this function, mainly in mice. And this is just a summary of that work. And what we know about what we call adult hippocampal neurogenesis is it occurs in the dentate gyrus. It's tightly regulated. it is important for learning and memory, and especially pattern separation. So what is pattern separation? This is the classic example we always give is you park your car every day roughly in the same location, but it's always a bit different. Maybe it's one floor up, one floor down. Sometimes you're close to the elevator, sometimes you're close to the staircase. And this is a very similar memory, and you have to remember that when you come back to the garage at the end of the day, where do I have to go? And this is one of the functions in aging that you lose very early on, because for the brain, it's really difficult to make that distinction. If remembering if you parked the car on the street versus the garage, that's easy, but where on the garage it was. And this is where these newborn neurons in the hippocampus can help you. And we also know that everything that is decreasing adult hippocampal neurogenesis decreases cognitive function and vice versa. And here I would like to make a small loop back to the mental health benefits. Stress is one of the most important factors that reduces adult hippocampal neurogenesis and could be one of the factors why in stress situations, especially in chronic stress also, cognitive function is decreasing. And everything you can do to improve your adult hippocampal neurogenesis, including exercise, certain changes in diet, aging is something we can't change. It's going down there. Some environmental, like noxious stimuli, if you avoid them, improves your cognitive function. So what about humans? This is histology, where they used complicated stainings to kind of mark these neurons in the brain. And then they looked at, these are different Braak stages in Alzheimer's disease. And you can see the more advanced Alzheimer's diseases, the fewer newborn neurons you have in the brain. And this is a different technology, which we call single-cell sequencing. So you can take the brain, dissociate it in single neurons, and using a technology where you can look at all the genes that are expressed in every single neuron at that time. And then you can figure out which are which cells. And again, it's a lot of figures, but the one interesting or exciting thing is what they did find is they find these immature granular cells, these newborn neurons in control brains, or like healthy age-matched controls. They also find them some in the Alzheimer, like humans with Alzheimer's disease, but it's going down. And then if you look at the function of the neurons, it's also going down. So this is a very important finding, again, that these might play a role. There are not many of them in there, but whatever they're doing, they seem to be still important. Unfortunately, we just don't have any imaging technology that I can take a picture now of your brain or take a blood sample and tell you, this is how many newborn neurons you have left, and you do an exercise, you take a medication, now they're getting better. So this is something we are still actively working on to get to this point. So I'm going to transition, talk a little bit about Alzheimer's disease. You probably have heard about it. It's a major neurodegenerative disease. Actually, in fact, it's the most common form of dementia. Many, many people are affected. Given that we have an aging population, the problem is increasing. And what has happened is you're literally shrinking your brain. So this is a healthy brain. We have the white matter and the gray matter, and you can see how the brain is shrinking. There are now, as you probably know, some drugs that are approved. Some were already taken off the market. Some are coming up. So there's some hope that we might get disease-modifying treatments, but we're still in early stages. And some of the ones that are approved have very severe side effects. So as physicians who have to make these decisions with your patients, you know it's not an easy decision to tell who should take them and who should not take them. And on a molecular level, what is happening is you have these amyloid plaques of aggregated amyloid beta. The amyloid beta, it's a normal protein that you have, but for reasons that are not completely understood in the aging process or with people with specific risk factors, they can make these sticky plaques in the brain. Tau, another important protein, can get hyperphosphorylated, also gets kind of sticky, makes these tangles. And what that leads to is you get an increase what we call neuroinflammation. So the other cells in the brain, apart from the neurons, microglia, astrocytes, they are trying to get rid of this substance, and they get very aggravated, what we call the neuroinflammation. And that actually can lead to the loss of synapses, the connections between the neurons, or even the neurons themselves. So some people say they die through friendly fire. So we are now understanding that reducing neuroinflammation, for example, by exercise, it could be a very promising mechanism. And there are also other pharma companies that are trying to target, either microglia or astrocytes in this process. So that's been a pretty new area of research. So coming back, exercise, does it work in Alzheimer's disease? In mice, can we use these mouse models? And the answer is yes. I show you two different data because that might be going a bit into the details, but Alzheimer's disease is a very complex disease. It's not that there's just one thing that's wrong in the mice and we can just easily model it in the lab. What we usually do is we use mutations in genes that give you the familial form of Alzheimer's disease, the one that can get inherited through generations. And then these mice develop Alzheimer-like symptoms. And these are 2 different mouse models where, again, exercise shows that the cognitive decline in these mice is more slowly. So we can use them to study the disease process. And the other part, which I just told you, we don't like these amyloid plaques. These are these brown spots here. And again, exercise reduces the number of plaques, again, in a dose-dependent manner. P-tau, the other, let's call it bad protein. These are basically non-transgenic control. These are the normal mice. You get an increase when you have the mice that get the Alzheimer's disease, and if you exercise them, it gets significantly reduced. And the similar is happening for neuroinflammation. Again, exercise intervention has a really nice multimodal way to address the pathology in the brain in Alzheimer's disease. And this is some very interesting observation. When you think about exercise, I've mentioned it's so complex, it does many things. Why does it do these things to the brain? And an original part of thinking was like, oh, maybe there's just things in the brain that are happening, synaptic activity, brain activity, and this is what's doing it. However, this data shows you that probably there's something systemically happening, like when you move your whole body, when you move your muscles, that's important. So there's what we call an enriched, I told you we can use these running wheels to exercise the mice, but there's something that's called enriched environment. So you have a cage and you put into the cage toys and other things the mice can interact with. And researchers have seen that this improves cognitive function in the mice. The problem is, usually there's also a running wheel in it. So what they did is they took out the running wheel here and left it in here. So when you have a control mice, this is, again, the black dots are the newborn neurons that you're making. If you start running, there are a lot of them, really nice increase. If you have the enriched environment with the running wheel, you see the same thing. If you take out the running wheel, even though you still have all the quote-unquote Sudoku puzzles for the mice in there, you actually don't have that strong effect anymore. So there's something about probably moving your muscles, changing the blood flow to the brain, changing your metabolism that is helping with the neuroplasticity you need to improve brain health. And this is showing neurogenesis, but similar data has been shown also for secretion of BDNF or cognitive function. So just to kind of wrap up here for that part is, what are all the things, how you improve, which is the neuroplastic mechanisms in the brain that are happening with exercise. So I told you can grow new neurons. I told you can reduce the neuroinflammation. Just the normal aging process will increase neuroinflammation, and you can combat that by doing exercise. Synaptogenesis and synaptic plasticity, meaning like either making new connections or maintaining strong connections between neurons. Exercise can help with that. You increase the blood flow to the brain, which might be in part why the hippocampus gets bigger, and all of this together helps with improving cognitive function. So kind of coming in, what our lab is really now interesting is, like, what are the molecular mediators? I told you all these nice data. We know it works in the brain. We know it works in mice. We know it works in humans. We also understand on a cellular level kind of which processes you have to push, but we want to know which are the molecules that we then can target to make a therapeutic, kind of similar that, if we understand how you get high cholesterol levels, we can, develop drugs to inhibit these pathways. So that's the same idea we have. How can we do this in exercise? And the idea or the research area we are in is myokines. So myokines are small natural peptide hormones that are coming out of the muscle when you exercise. They have been described, and people already know that they can travel to the bloodstream and have different effects on different organs, pancreas, fat, bone, liver, circulation, and also the brain. And this is part of the work that we did, that there's this muscle-brain crosstalk. And the prime candidate we are looking at is called Irisin. It's a small natural hormone. It's like, if you think about it in scientific terms, it's pretty tiny. It's 112 amino acids. With glycosylation, it runs at 24, 25 kilodaltons. It's 100% identical between mice and humans, which makes it a great gut target. And the way irisin is produced is you have it expressed in skeletal muscle. You also have it in the brain. And then in exercise, it makes a precursor hormone that's called FNDC5. And then this part that is sticking out of the cell membrane can get cleaved off, and that's actually the irisin part. And then it's in the bloodstream, and we and others have shown that it can travel in the blood, and actually exercise increases the blood levels of irisin in mice and humans. And again, the evidence that we see irisin induction is stronger, or the strongest in aerobic exercise. So, and this was the first paper showing this. When I started my work as a postdoctoral fellow in Bruce Spiegelman's lab, they show it comes out of the skeletal muscle, it can travel to the fat, it has some metabolic effects there, so it can help you, it's part of the reasons why exercise is good for your metabolism. But as I had seen is that it was very highly expressed also in the central nervous system, so we came up with this idea. Okay, we know exercise is good for the brain. We know irisin is an exercise hormone and has beneficial effects. Is there a role for irisin actually in exercise in the brain? And long story short, this was the publication that came out of it, where we showed for the first time that there is a direct link from irisin to these BDNF pathway in the brain in exercise. So BDNF is one of these neurotrophic factors that can help, for example, grow you new neurons. And there was a follow-up study from a Brazilian group where they looked at FNDC5, irisin also in Alzheimer's disease. One problem with both of these studies is we, at this time, were only able to look at FNDC5, the precursor protein, but not directly at irisin. So why is this important? Why should we care about this? So the reason why we care about this is if you're a pharma company and you are interested in making a drug out of it, you want a small peptide hormone that you can target versus working with a very long type 1 membrane protein you don't even know how to produce. So in my work in my own lab, we kind of really dove into how does irisin work. And we showed that it's actually a critical regulator of cognitive function using Alzheimer mouse models. And showing that it changes neurogenesis and reduces neuroinflammation. So this is the data. Again, I'm going to go through it a bit quickly. If people have more questions, I'm very happy to delve into every single molecule and subfigure later. So again, what we did is we generated mice that, like, you can do this. You can take out the irisin hormone out of mice, making knockout mice, and we can show that if you do that, these mice, when you do them an exercise intervention, the wild-type mice learn faster, but the knockout mice, there's no difference if you exercise them or not. We then looked at the newborn neurons, and again, you can see that not having irisin was giving them an abnormal function. We looked at how the neurons looked like, again, using very sophisticated reporter gene technologies where you have to inject viruses directly into the brain, into the region where the stem cells are, and did a lot of, my postdoctoral fellow, Rached, did a lot of really sophisticated analysis, and the bottom line is the way the neurons develop is abnormal. They have too many branching where they should have less, and they have not enough synapses where they should have more. And if you then use a trick to kind of sort these neurons out, when you mesh up the brain, we have a trick how we can only get the newborn neurons out, and we do transcriptional sequencing to look at the gene expression. Again, we can see there are huge differences between having irisin in your newborn neurons or not. Then we went on to look at the Alzheimer mice. So we crossed our mice to an Alzheimer mouse model. So these mice have Alzheimer, and they either have or don't have irisin. If you don't have irisin, and you do a task where you have to distinguish between two contacts, the wild-type mice can still do that, or the Alzheimer mice that have irisin can do that, but the mice that don't have irisin cannot do that. So their cognitive decline is accelerated. Then we did the opposite. We did the therapeutic experiments. We used gene therapy approach to give back irisin. We actually injected it into the tail vein. It goes to the liver, and then the liver pumps out irisin. You can find it in the bloodstream. You get a nice increase in the plasma irisin. And then we did the Morse water maze, and you can see again the the mice performing better in the Morse water maze, so they have better cognitive function. And the point I'd like to make is these APP mice, so this is again these human familial mouse models. People have maybe heard about these genes that are important for Alzheimer's disease. These mice already had the plaques and the neuroinflammation, the cognitive deficits when we treated them. So this was a therapeutic, not a preventative trial. And to make us and the reviewers feel better, we repeated that in a different mouse model that was even more severe. So these mice not only had amyloid plaques, gliosis, cognitive deficits, they also had the neurodegeneration. So neurons were already dying. We treated them with irisin, and after this intervention in the Morse water maze, they performed better. So this doesn't mean that you give the mice irisin, they're going to be reversed completely back to normal, but it shows that you can slow down the cognitive decline. And again, if you think about patients, if you can extend disease-free lifespan even for months or years, that's a huge boon for life quality. And again, going back, how does irisin do that? We did molecular analysis looking specifically at the cells that are part of the neuroinflammation, and they looked better by, if you looked at the morphology of the cells, that looked improved, or if you did, again, a gene sequencing analysis to look at the genes that are expressed, they showed that they were less activated. Lastly, something that researchers are always interested is if there's a secreted molecule, where does it bind to? And previous work from Dr. Spiegelman's lab is where we collaborated with showed that there was a specific irisin receptor called an alpha V beta 5 integrin receptor. And then we showed that in a CNS culture model where we took out these adult hippocampal stem cells and differentiated them and used this super resolution microscopy where we can look at every single molecule in the cell, we can show if we put irisin on these cells, these are the red dots, that they actually can stain, that they bind to the green dots, which is the integrin receptor. So this is what we show here. So irisin can bind to integrin receptors, or more specifically to a specific subtype of integrin receptors. And then if you look which cell types express the integrins, it's actually the astrocytes and not the neurons. There are no microglia in it, so we don't know about that yet. This is follow-up work from Dr. Choi's and Rudy Tanzi's lab. Rudy Tanzi, maybe people haven't heard of it, very big in the Alzheimer field. They used a human 3D culture model, so like we call it the Alzheimer in the dish model. You can take human cells and they develop, Alzheimer-like phenotype, and they treated these cells with our irisin, and they can show that there was less of these amyloid plaques, probably through this ERK-STAT3 signaling pathway, which again, if someone wants a detailed walk through this signaling cascade, I'll be happy to do this later. And then kind of switching away from Alzheimer's disease, Parkinson's disease, another very important neurodegenerative disease. And in collaboration with Ted Dawson, he's over at Johns Hopkins. He is an expert in Parkinson's disease. Irisin, we tested it in his model. So what they do is they inject these preformed alpha-synuclein fibrils, like the toxic Parkinson protein, directly into the brain of the mice. So they get Parkinson-like symptoms, which means in the substantia nigra, if you remember that, the dopaminergic neurons die the same as they do in humans, and then they start to get motor function problems. And if you give irisin, again, using our AAV irisin trick, you can show that you actually lose less of the dopaminergic neurons. And if you do like different motor function tests, the pole test where they have to climb up a pole so you can see how good they can still hold onto, or you measure the grip strength with a little grip strength meter, the mice that get irisin actually lose much less muscle strength, or they preserve the muscle strength, which is quite exciting that it seems that whatever irisin is doing, it's not specific to one neurodegenerative diseases. There is like some mechanism that translates, and neuroinflammation is certainly one of them that could be there. Oh, okay. I thought I had one slide in there more, but that's okay. We also started testing it in a different mouse model for Alzheimer's disease, tau mouse models, and we can also show that irisin was beneficial in the tau models. So kind of switching gears a little bit, I talked a lot about irisin, which is our favorite hormone, our prime candidate, but there are other techniques we can now use to try to unravel how does exercise actually affect brain health on the single cell level, and that's something that we did. So we had, again, wild type mice and the Alzheimer mice. We did them with and without exercise. We again show that the exercise nicely improved cognitive function. We did histology to show that they had more neurons. We showed histology to show that they have less plaques and less neuroinflammation. And then what we can do is we can dissociate this part of the brain where the stem cells are, and we do the single cell sequencing. And you can see that we find all the major cell types that we need in there. We find the neurons, we find different glial cells, astrocytes, microglia. OPCs are the precursors for oligodendrocytes, very nicely there. But we also found, what we were very excited about, we found cells of the neurogenic lineage, meaning neuroblast 1, 2, and here in green, the immature neurons. This is just to show these are the marker genes that we can use to identify these cells. And there's a lot of work in this paper. It's still in revision, so I can't show you all the data. But one thing that we use the data for is to understand how gene expression is changing in the brain. So the idea is there are genes that if you are going from a healthy or from a wild-type mouse to an Alzheimer mouse, they're downregulated. That's part of the disease process. But then with exercise, you can bring them up, or vice versa. They're going up with Alzheimer's disease, you can get them back down with exercise. And this is what these graphs are showing, and we were able to do this for every single cell type. There's not as much here because we didn't get that many cells, but if you look at immature neurons, mature neurons, microglia, very excitingly, astrocytes, all the genes that you kind of see that are skewing this way or getting red, these are genes that are dysregulated in Alzheimer's disease, so they're part of the Alzheimer pathology, but you can get them back on track with exercise. So in the paper, of course, dissects all the different genes and what they're doing, and we show proof of principle, how they improve pathology. So this is kind of our new exciting work, which we are working on to see if we can find more factors. So question left, if you, either for yourself or talking to your patients, how much exercise do I need to do for my brain? I will quote the guidelines here from the CDC and the American College for Sports Medicine, minimum of 150 minutes a week of moderate intensity PA. You have substantial health benefits, meaning if you get there to this part, you can increase the benefits if you go to these like 300 minutes a week of moderate intensity. They also always have a substitute that if you, instead of doing moderate, you can do intense exercise, you can do fewer minutes, and you get more benefits from that. And then again, importantly, also do muscle activity or muscle strength activity training. I think that's also very important to make this, relay this to your patients. This could be yoga, it could be weightlifting or other things. Kind of like my take home on this, like the kind of like what I would like to say is, as easy guidelines, more is better. Like, I wouldn't fuss too much about how many minutes it was, but as a guideline, if you can do more, it's better. Every step counts. So meaning if you park the car and you can walk your way inside versus, taking the elevator, if you can, instead of, taking the shuttle from the train station to get to your workplace, you can walk, that counts. If at home you can do, if you're raking the leaves instead of using the leaf blower, every little thing you can do to improve or increase your physical activity, it does count. And I think I said it already in the beginning of the talk, but again, going back to the patients that are already struggling with fragility or declining health, I think that's a very important factor to get the motivation on, because if you're young and healthy in your 20s and 30s, sometimes it might be hard to understand how it is when you're not, maybe you had an injury and you bounce back within a week and you're back where you were. The older you get, or if you have underlying chronic conditions, it's not that easy anymore. Another thing I always like to say, stick to what you can stick with, meaning a lot of data is on running exercise. But if you are someone who does not like to run, and then you don't do it, it's much better to find an alternative. If it's vigorous walking, if it's hiking, maybe you have a friend and you like to bike, and it's much better that you do bike tours on the weekend than trying to figure out how to force yourself to go to the gym on the weekend. And also thinking again, my previous speaker talked about diversity and opportunities. For, some of us have, like free gyms where we get a privileged membership in right next to our work, and it's not a problem to choose between, I don't know how many fitness classes and make it work, but that's not the reality for everyone else. So again, when you counsel patients, also get an understanding of what is feasible for them. And maybe if it's only the yoga class on YouTube they can do a couple of nights a week, because they can't go to the nice fancy yoga studio and sign up for their class pass, better than not doing anything. So that's kind of my take on that. Okay, so then how do I know my brain is healthy enough? So my colleagues at MGH came out with this great brain care score, where you can look at different domains in your life to see how healthy you are. Some of that I had it, I think, highlighted. Importantly, there's exercise on there, there's sleep on there. There are also other things in there like, general health, is your blood pressure under control? Do you have type 2 diabetes? Is it under control? Do you drink alcohol? How much do you drink? Smoking? And these are all things, again, that's why they made it as a scorecard. You can print it out, give it to your patients, or, if people like the electronic version, you can do a little quiz, click yourself through, and then it will also tell you, oh, these are areas where you can improve. So I think this is good for, for the counseling part, and maybe also doing then the follow-ups if you do your regular checkups to kind of have a discussion. Hey, we talked about this and that. Were you able to make some improvements in there? And then the last question, when can I get the exercise pill and stop worrying about actually exercising? The short answer is for that, or let's start with this one. So there are, apart from our research where I talked about Irisin, which, due to my very biased opinion, which is a great candidate to become an exercise therapeutic, there are other molecular secreted factors that people have looked at. When you think about developing actually a drug on it, there's a long way, and some things that work great in mice might not become a drug. For example, a lot of mediators that improve cell proliferation, for example, give you more, proliferate the blood cells in the brain, the blood vessel cells in the brain which you want, or the newborn neurons which you want. If you give them systemically as a pill, they might also stimulate cell growth in other areas of your body, which you probably know is then could lead to like tumors or cancer. So that's one of the things. You have other medications that have severe side effects if you give them not with a viral vector, but as a different. So we have a lot of candidates in there. Some of them are better candidates than others, and we'll see. However, what I would like to caution about is exercise does many things to your body. So I mentioned it. So it's not just the brain health benefits. It's about the muscles. It's about the metabolism. And even if you think about the brain part, I talked about cognitive function. We already heard from, we're hearing about the mental health. So the idea that there is just one mediator that is gonna give you, bulks you up, gets rid of your diabetes, makes you smarter, and reduces your anxiety levels. I think that's going to be a lot to ask, and given that exercise has these pleiotropic effects on all of that, I would advise to the people that can exercise that they should rather exercise to get all the benefits they can get. However, flip side of that coin, if you think about aging population, my father has a chronic disease and is frail, so these patients might not be able to exercise to the level that they would need to actually get the cognitive benefits. So if you need to run, a mile every day and you barely can make it to 2,000-3,000 steps a day, you just can't get there. So I think the same way, or what also might be true is that even running 1 mile at the point when you're having, starting to have cognitive decline doesn't boost your irisin levels that much as you need, or your catepsin levels, or your other levels. So what we then can do is, when we give it as a drug, we can boost them to a pharmacological level, like more than what exercise intervention could do. And so for these patient populations, I think developing drugs based on exercise mediators is a very promising direction, because as far as I know, there's none of them in the clinical trial yet. So this is a completely new category of drug, candidate directions that we are that we are taking. And so maybe, hopefully, some of them come through in the future, which would be really exciting. All right, so to kind of summarize, exercise improves cognitive function and prevents dementia. I have shown you that. We have discussed important cellular mechanisms of these neuroprotective effects in exercise: neurogenesis, increasing blood flow, angiogenesis, synaptic plasticity. And then we have talked about the exercise hormone Irisin, which is a crucial molecular mediator of cognitive benefits to exercise, and the potential drug treatment for Alzheimer's disease or Parkinson's disease. And I would like to end with an acknowledgement from the people in the lab that did the lot of this hard work. Joanna de Rocha, one of the postdoctoral fellows, Luis Moreira, Arif, Rashid, and they were supported by a great team of graduate students, medical students, and research assistants. We have a great team of collaborators on the bioinformatics side, on the exercise side, on the Alzheimer's side, and obviously, without the funding, we could not do our work. So I'm very grateful also to the NIH and different foundations that have supported my work. And I'll be very happy to take any questions, and I think we also have some time.
What a wonderful job, Dr. Wrann. Honestly, very engaging information. I got to start reading a book while I exercise, make sure I hit those metrics. I'm going to shoot for that 300-minute one.
That's good.
Otherwise, let's get the questions going. If anyone has anything, I see some people getting up here, so we'll wait a little bit. And then, as people are getting up to ask some questions, for those of you who weren't able to ask all your questions in the earlier session or moving forward, please feel free to email our foundation. We'll send the questions to the speakers at info@abkf.org. It's the acronym of the foundation, abkf.org. And that's the info part. Okay, so otherwise, let's take our first question.
So, do we have any research or evidence that shows that cooperative exercise has more of a...
Which one?
Cooperative exercise, so more team
sports. Yes. Okay, I'm just going to repeat the question I was asked so that the live audience also gets them. So the question is there evidence that cooperative sports, team sports, have more effects than individual sports? Very good question. I think on the cognitive side, not as much. Most of the focus was, as I said, is like, do you improve your cardiorespiratory fitness? But I do know, for example, for Parkinson's disease, when you look at, slowing down, dancing is one of the exercise intervention that is very often recommended for two reasons: the social component, and also if you already have people with a bit of impaired stability, doing dance, like couples dance can help, and also the rhythmicity that you have. But I think it would be interesting to see if the social component in itself, how much of an effect it has. We do know that social isolation definitely does have detrimental effects. So I would say it has a part of it. I don't know if we can quantify that part yet. So good question.
Thank you.
Fascinating work.
Oh. There goes the mic.
It was so great.
I thought I was the one who's doing them with the mic drop in the end.
I got a quick question about the role of nutrition.
Yes.
The animal studies that you've done, where they all exercise at the same amount, same type of food that they have, and whether or not in general, going back to humans, and I'm not sure whether they can extrapolate on that, whether nutrition has any factor.
Yeah, very good question again for the live audience. The question is on the impact on nutrition, on brain health, and especially cognitive function in animal studies in humans. Nutrition does have a very big effect. So the studies we did to take out the nutritional part, the mice were fed what we call a chow diet. So that's the regular diet where they don't get too obese on. However, if you put mice on a high-fat diet, either high-fat diet or high-fat and high sucrose diet, that will lead to a decrease in cognitive function. So what happens is you get a lot of neuroinflammation, you get a reduction in neurogenesis, And some studies have shown, again, if you then get the mice to exercise, you can reverse some of these effects. Then it's always a bit difficult if the mice exercise. It might also change, they're going to burn more energy. So how much of that is a bit of the weight loss that's happening versus the exercise? But yeah, healthy nutrition is a very important factor. And on the brain health scorecard, BMI or like body weight is one of the factors that is monitoring. Sometimes I get asked about specific supplements that are the brain boost. I don't think there's one diet thing. I mean, if there would be one, I think it would be prescribed by your doctor and everybody would know about this. As a general recommendation, what we already know is like if the diets that can, help you to keep your weight down. So more plant-based, less high fat, less high sugar. Usually that means less processed foods because, they're the ones that contain that. So yeah, maintaining a healthy body weight goes a long way to reduce the risk factor to develop cognitive decline in either aging or dementia.
Thank you. That was really, really interesting. So I'm wondering if anyone has looked at the effects of Irisin on or the role of Irisin in some of the more emotional effects of exercise. So raising mood, reducing anxiety, things like that.
I'm looking to my colleague if I get a thumbs up to comment on that. I don't know if I see him. No? It's like we are investigating it, and please stay tuned. I don't want to give anything away that without his permission. But yeah, it's an obvious direction to look at. So the question, yeah. And maybe find me after the talk, and I can point you towards my colleague, and maybe he can what he's happy to share already publicly.
And if I could ask one more question. So I know it's known that, or there's evidence in preclinical models that autism seems to involve inappropriate, maybe excessive aging, and autistic individuals have very interesting differences in movement pattern and
Yeah.
a lot of excessive movement. Do you think there's any link there between maybe excessive movement, driving, excessive adult neurogenesis through perhaps your pathway?
Yeah. So very good question. The question is there a link of excessive movement and increases in adult hippocampal neurogenesis? And one example that was named is autism spectrum disorders, where you have, I guess, hyperactivity, excessive movement in there, and how that relates to mouse models. So I'm not an expert in autism disease, so just as a disclaimer. However, there's an interesting mouse study that might answer your question. So they did what we call outbreeding. So they had mice and they kept breeding them. And then what starts happening is you're going to have some mice that very naturally don't move a lot. So if you have them with a running wheel, they almost don't run it, so they're sedentary. Then you have the one kind of in the middle. That's the one we usually have, the regular wild type mice. They run 5-7 kilometers. And then you're going to get the hyperactive mice that are running 12-15 kilometers at night, which you would call excessive hyperactivity. And there we then see also these mice start to get problems because you are on the other side of that spectrum. So as most of the things in science or biomedicine, you want to be in the middle. And these are the mice that actually, they can't sit still. Like they do have to do that. They get also, I think they have higher stress levels. They have much higher levels in their motivational activation system, what keeps them running. And you didn't ask that question, but maybe linking that back to humans about what about people who do ultra marathons or super, active sports? So sometimes I get asked about, NFL football. Again, if you're going beyond a certain physiological level, doing more is probably not giving you benefits anymore. I don't know if anyone has done studies with autism and running wheels in mice, and I don't know if it would make it better or worse if they run more and it gives them an outlet to release the tension, or if you then have a mouse that is basically constantly running. I have a friend who's a colleague at MGH who's doing autism spectrum disorder, so I'll ask her about that. But yeah, thank you very much.
Thank you very much for your presentation. You talked near the end about the ideal of having a minimum of 150 minutes moderate exercise ideally 300, so they can get the Irisin release. And looking at people that have differing abilities,
Yeah.
is there suggestions, considerations, insight into types of exercise that is accessible? Like, for an example, I'm seeing on social media a lot about bouncing on the rebounder for cardiovascular and lymphatic health. And I'm just curious if you have any insight on that, so you can make suggestions on what can be accessible to reach those goals.
Yeah, very good question. So the question is, people of different abilities for them, like maybe the traditional 150 minutes per week through running exercise might not be accessible. Could there be alternatives? One was suggested, the bouncing on there to get your cardiovascular system going. Specifically for Irisin, we haven't done any studies yet to kind of go through all the different modes. As I mentioned, most of the studies points towards that everything that improves your cardiorespiratory fitness, you will have brain health benefits. So the good news is also if you start with someone who has a very low aerobic capacity, even if they do not, quote unquote, much exercise, that's probably already challenging, right? Like if you have someone who barely does 1,000 steps, if you get them to walk, 2,000 steps, they're going to be out of breath. They're going to be sweating. For them, that was a huge, effort. So I think that's the good news. So I would encourage patients not to be too stringent about thinking about the 150 minutes have to be run on a treadmill. But what are things that gets your heart pumping? I think that's a good measure to get there.